Hey guys, sorry about the whole breathiness of the last couple lectures. I just listened to some of it back and I heard that I was breathing in the microphone. So sorry about that. I moved the microphone further from my mouth, so hopefully that won't be an issue. Okay, let's pick up where we left off. Understanding number 4. Phospholipids are ambipathic lipids formed by condensation from 2 fatty acids and 1 glycerol. So phospholipids are used in cell membranes like we said. And these guys have 1 glycerol molecule head and 2 fatty acid tails. Now, that head is hydrophilic, and both the tails are hydrophobic. Now, when we say hydrophobic and hydrophilic together, meaning we have a molecule that's both, one part is hydrophilic and one part is hydrophobic, that's ambipathic, just like ambidextrous. Like when you're ambidextrous, you can use both hands. Well, ambipathic molecules have both hydrophobic and hydrophilic properties in different places. It's both. Triglycerides, on the other hand, are molecules that are components of fats and oils, and these guys have 3 fatty acid tails instead of 2. So they're just like phospholipids, except for that they have 3 tails. So here's our glycerol head. How does glycerol bond with these fatty acids? Let's see if you can predict how many water molecules are going to fall out. And let's see if you can tell me the name of this chemical reaction. So this is a condensation reaction, right? Dehydration synthesis, and it's an anabolic reaction. We're building stuff. Now let's see how many water molecules we get. 1, 2, 3. There we have our triglyceride. So how are we going to break this down? Well, the same way we broke down all our other molecules. We're going to use water, right? This is hydrolysis. It's catabolic. We're going to add that water back in to break those fatty acid tails away. So let's compare. Phospholipids are used in cell membranes, right? They have a glycerol head and 2 fatty acid tails. Meanwhile, triglycerides are used to store energy, right? And they have a glycerol head and 3 fatty acid tails. A3. Lipids are more suitable for long-term energy storage in humans than carbs. All right, so lipids, right, formed by fatty acids, they're more suitable for long-term storage than carbohydrates because they store more energy in the same amount of weight. Remember, energy is stored in those bonds between the carbon and hydrogens, right? So think of all those carbon hydrogen bonds in the fatty acid tails. There's a million of them. This also aids in heat insulation. So if you take a look at this table right here, we have carbohydrates, we have triacylglycerol, and we have protein. This is the dry weight. So if you made it into a dried powder, right? That's what it would be. That's the weight in grams or kilocalories per gram. So this is how many calories per gram is in each of these things. How much water weight there is in these molecules when they're not in their powdered form or dry form. Kilocalories per wet weight, we don't really care about that. We don't really care about either of these two. What we really care about is the total amount of energy stored. This is what we care about. So, total energy stored in kilocalories. Carbohydrates store about 840 kilocalories per 4 grams of dry weight. Our fatty acid, our fat here, it's not really fatty acid, it's fats, right? Triacylglycerol, this is a fat, stores 135,000 kilocalories in a little more than twice the weight, right? So if you doubled this to 8, so it was comparable to 9, this would be 1640. 1640 compared to 135,000. It's ridiculous. And then there's protein. Protein does store a bunch of energy. You get about 24,000, but we don't really, the purpose of protein isn't really energy storage. The purpose of protein is building bigger molecules, amino acids building functional molecules. So protein's not really an energy thing. It's more of like a muscle recovery thing. So, long and short, fats, lots of energy, insulation, sea doggos are cute. All right, let's talk about your health. So, there's lots of fats out there, and there's lots of claims about the different types of fats out there. And we should take a minute and talk about coronary heart disease. So, coronary heart disease is when plaque builds up in the coronary artery. And the coronary artery is the artery that actually delivers blood to your heart so that your heart can function. Not coronary artery like the artery from your heart that delivers blood to the rest of your body, not the functional part, but like your heart is a muscle, so it needs oxygen, right? So the coronary artery is the vessel that moves blood around the heart for the heart's supply. When this is clogged, right, you're reducing the amount of oxygen that can be delivered to your heart. If your heart doesn't have enough oxygen to maintain beating, that's called a heart attack. So, saturated fatty acids, which are found in dairy and meat, remember, these are animal fats mostly. There's a strong positive correlation between saturated fat intake and coronary heart disease. If you just look at this, right? If you just look at this line, it looks like, okay, the more cholesterol and saturated fat you eat, right, the more incidences of coronary heart disease you have. That seems pretty simple. Except for there's a problem. There are a couple of places, like France, where their index, right, how much fat they're actually taking in is pretty high. But they seem to have a much lower rate of coronary heart disease than somewhere like Finland, who doesn't really have that much more fat intake, but their incidences of coronary heart disease are like way higher. So this indicates that there's something else going on here. It can't just be saturated fats that are the problem. This is a good example of why you have to remember causality is not necessarily implied with correlations. So, correlation does not imply causality. So, some populations have diets that are really rich in saturated fats. Think of like the Maasai tribe in Kenya, but they have like zero coronary heart disease. It's crazy. So, there's more to the story that we're still trying to figure out. A recent study looked at trans fat consumption. Now, these are fats with trans fatty acids, right? So they have the hydrogens on opposite sides. These show a positive correlation with coronary heart disease, right? In the presence of other risk factors which prove negative. So it's likely that this relationship is causal. So here we have a study that looked at all different types of fats, right? Instead of just looking at saturated fats, we looked at polyunsaturated fats, monounsaturated fats, saturated fats, right? And trans fats. And this pink line is just a zero line to show you zero goes across, right? So if we look at the percent in coronary heart disease as related to the content of each specific kind of fat, you'll see diets high in polyunsaturated fats tend to have very low coronary heart disease, right? It's negative. Monounsaturated fats tend to be the same thing. Saturated fats, you do get some increase in coronary heart disease, but trans fats show the largest increase in coronary heart disease. So, to make this clear, here we have a study that looks at just saturated fats, and we found that there is a general positive trend. The more saturated fat index, or the more saturated fats you eat, right, the more likely you are to have coronary heart disease. Except for that, there are some places that have high saturated fat indices, right? But they don't really have a lot of coronary heart disease. Well, these guys do. That means there's more to the story. There's got to be some other fat. So this group is eating lots of saturated fat, but they're also eating another kind of fat that's causing this. These guys are eating lots of saturated fat, but they're not eating that other kind of fat. That other kind of fat is trans fat. It's important to understand how to analyze claims. If we had just looked at that first study, we probably, well, we know we didn't get the whole picture. So, whenever you're looking at the results of studies, there's a couple of things you have to ask yourself. How big is the survey size, right? Are you getting an accurate picture of the population? How even was the sampling? Are you representing everybody? If it was uneven, are you adjusting your results to account for some of that? Were the measurements of lipid intake and coronary heart disease rates reliable? Were the measurements reliable? That's kind of like analyzing the method, right? How did they do the experiment? Analyzing the results is a little bit different. Now, the results of the first study showed a correlation between saturated fat intake and coronary heart disease, right? That's what they were implying. But in reality, they were measuring more than just saturated fat intake. They were measuring other types of fat that they weren't accounting for. Were there differences in coronary heart rates, coronary heart disease rates, and lipid intake significant? Yeah. Look at Finland and France. Those were two countries that had similar intakes of saturated fat, but the results of coronary heart disease were totally different. That's a clue that there's something going on. Statistical tests, when we talk about stats, we'll talk more about that. How reliable is their R value? What's their type 1, type 2 error? And how variable was their data? In that first study we looked at, there was a lot of spread. And that's also a good indication that there might be more going on that we're not capturing. Okay, next is determining your body mass index. So a nomogram, that's what this thing is a type of chart that can be used to find your body mass index. To use it, you're going to mark your weight on the left, so whatever your weight in pounds is. And then you're going to put your height on the other side, right, in inches. And then you draw a line between the two, and it crosses this central axis. And that tells you your BMI. So, oh, wait. Okay. Using the nomogram, what is the body mass index of a patient who weighs 130 kilograms and is 65 inches tall? Okay, so I'm going to find 130 kilograms on the left, right? 130 kilograms, so that's right here. Why isn't it drawing? Draw. So 130 kilograms right there. 130, and they're 65 inches tall, so inches, 60, 65 inches tall. Okay, now I use a ruler to draw a line between those two dots. There we go. And where it crosses, that is the body mass index. Let's see, I think our answer came to 41, 42, 41 and a half. 41 and a half would be the body mass index for someone that's 65 inches tall. Okay, so now what I want you to do is I want you to pause the video and I want you, let me take these off. I want you to see if you can calculate your own body mass index. And if you can pause the video and you can hold a piece of paper up to the screen. And I want you to, for your answer, I want you to write down your weight and your height and calculate your BMI for me. Okay, one more thing we should talk about before we go. Steroids. Steroids are lipids. A good example of a steroid is cholesterol. And we talked a little bit about this in the very, very beginning, right? So a lipid is a glycerol head and a fatty acid tail. But in between the glycerol head and the fatty acid tail, there's a bunch of carbohydrate rings, right? So the hydroxyl group makes the head polar and it sticks out towards the outside of the cell, the water part, and hydrophilic. And then you have the carbon rings, and then you have the fatty acid tail at the end, and those are hydrophobic, so they point inwards in the inside of the lipid bilayer, right? because they're attracted to, well, not attracted, they're repelled from water, so they want to be as far away from it as possible. Also, when we're talking about coronary heart disease, right? This is an artery. RBC is red blood cell, right? That cholesterol is going to build up, and fats are going to build up in the walls, which blocks the flow. Just in case the other stuff that we talked about wasn't very visual, it's just a visualization of like how heart attacks happen, right? You get like a traffic jam. Atherosclerosis is a disorder in which you've got that plaque growing in the artery, and there's actually a layer of skin or a layer of tissue. It's not skin, a layer of tissue that grows over that, right? So you're literally like growing a flap of tissue over that. And you can see here's this clogged artery. Big problem. If your cholesterol is over 240, that's considered high. If your cholesterol is between 200 and 239, that's considered borderline high. If your cholesterol is less than 200, that means you're doing good. Okay, at this point, you should be able to compare the properties of saturated and unsaturated fatty acids. You should be able to compare the structure of cis and trans unsaturated fatty acids. You should be able to outline the formation of a triglyceride, and you should also be able to outline why lipids are more suitable for long-term energy storage. If you can't do any of those things, make sure you go back in your notes, watch the lecture again, and of course, make sure you are reading the textbook. You are reading section 2.3 to go along with this lecture, okay? All right, I'll see you guys soon.